All right, hey everybody. My name, for anyone who doesn't know me, I think everybody does at this point, hi, Kopecky. Today I'm going to talk to you a bit about some work I did during my PhD, mostly focused on coral reef systems and how we studied changing disturbance regimes in coral reefs and developed some cool remote sensing tools coupled with AI to quantify the impacts and consequences of those changing disturbance regimes. And then I'm going to talk a little bit about how I'm taking some of those tools and topics and questions and applying them to other systems with my work here at EDS. So Jill Marie already pointed out that she doesn't know what one of these words means on the title. Don't worry, I'll explain that. I see you have like little colorful pipe cleaners. You can just put up a pink colored one if I say something you don't understand. I'm just kidding, I'm colorblind, it won't work. So just to set sort of a general ecological context here. What I generally focus on as an ecologist are the impacts of disturbances. And these are events we see throughout nature, like landslides or fires or floods or storms that can really dramatically change entire landscapes. But what I find really compelling as an ecologist is not the destruction and damage these events cause, but the ability for ecosystems to rebound from those events, which is a form of resilience. So there are a lot of different ways we can quantify disturbance. You can quantify the intensity of a disturbance or how much mortality was resulted from an event. You could quantify the time in between successive disturbance events or the frequency of disturbance, or something about what condition the environment is left in after a disturbance occurs. And so collectively together, we could refer to all these metrics as a disturbance regime. So over long periods of time, ecosystems can sort of become adapted to certain disturbance regimes, represented by this little red ball here. And once they become adapted to a certain disturbance regime, they become resilient to those disturbances and rebound continuously from them. But under global change, we're seeing that these historic disturbance regimes are being modified in different ways. Maybe the disturbances are getting more intense. Maybe the time between disturbances is shrinking, or the post-disturbance environment is being left in some different condition. And all of these changes can create misalignments between the disturbance regime an ecosystem is used to facing and the one it's currently facing, which calls into question whether or not contemporary ecosystems are going to continue to be resilient to modified disturbance regimes. So a lot of attention has been paid so far to studying the intensity and frequency of disturbances and how those are changing. But less focus has been put on this post-disturbance environment metric, which is what I'm going to focus on. Particularly in the way that it facilitates what we call ecological memory. So just like how we as humans and past things that have happened in our lives can guide our future behaviors, ecosystems also have a form of memory where past events can influence their current trajectories. And we think of ecological memory as having two different components. The first we call informational legacies, which is like heritable life history traits that can be passed on from communities before a disturbance into communities after disturbance. And over long periods of time, this can sort of create an adaptive response where ecosystems can become adapted to a new disturbance regime. The other component are what we call material legacies. And these are physical structures, living or dead, that persist in an environment after a disturbance occurs. And these can have really immediate effects on ecological processes that influence community assembly. And because of this more immediate effect, I'm going to focus on material legacies because these are the things that are really causing problems for ecosystems as disturbance regimes are changing and leaving behind novel legacies. That definition work for you, Julie? So I look at ecological memory through the lens of foundation species and the material legacies they leave behind. Foundation species are the organisms that create the actual structures of ecosystems. These are things like trees and oysters and shrubs and corals. And foundation species typically dominate their ecosystems in abundance or biomass. Therefore, when they die, their remains can be really pervasive as material legacies. And as disturbance regimes are shifting under global change, we're seeing novel material legacies being left in our environments. And one of the systems that exemplifies this best is in coral reefs. So here's a typical healthy coral reef where there's lots of structural complexity provided by these reef building corals that provide habitat for fishes and invertebrates. And historically, the disturbances that were sort of the main cause of coral mortality on coral reefs were tropical storms like cyclones or hurricanes that create big, powerful storm waves that act to break up coral skeletons and sort of remove them off the reef and leave behind this really flattened reefscape. And historically, coral reefs have been sort of resilient to tropical storms. But in recent decades, we've been seeing this rise of a different type of disturbance. It's marine heat waves that cause coral bleaching. While these also kill corals en masse, they leave behind a fundamentally different landscape after they occur, where all that structural complexity of the coral skeletons is left in place. And so this sort of raises a really clear question. How does that dead skeleton legacy affect coral reef resilience? And will coral reefs be resilient to heat waves just as they were to tropical storms? So first, I'm going to tell you about this technique that we developed to sort of accurately and efficiently quantify the impacts of marine heat waves using underwater remote sensing and AI-powered image analysis. Then I'm going to tell you about how we applied that technique to an experimental setting to ask or to answer the question of how do these dead skeletons affect community trajectories on coral reefs. And lastly, I'm going to tell you how I'm applying both that tool and trying to ask that same question across a more broad array of ecosystem types here at EASL. All right, so starting with this remote sensing technique. Speaking to a room full of remote sensing experts, I have to tell you that remote sensing is this really powerful tool that enables us to collect high resolution data over really large spatial scales. So it's an amazing window that's been opened into ecological exploration. But there's this bottleneck associated with extracting data from remote sensing products. There's just so much data packed into them that sometimes it can be sort of difficult to actually get the data we need out of them efficiently. But fortunately, with these advances in AI in the form of machine learning, it's helped to reduce that bottleneck in a way that we're able to efficiently and rapidly extract lots and lots of data from remote sensing products. So coral reefs, the study of coral reefs has benefited a lot from bringing remote sensing underwater. These are systems that are really structurally complex and they're just packed in with life over every square centimeter of the reef. And it's notoriously difficult to do anything underwater. Anything you do on land is 10 times harder underwater, not to mention breathing. So when you're down there, you're actually very constrained on the amount of time that you have to study these systems. And therefore, you're really limited in either the spatial extent that you can study effectively or the spatial resolution. Usually there's a big trade-off there. But by bringing remote sensing underwater, we're able to create these big digital permanent records of the reef that we can continue to draw data from. So I'm going to walk you through the different steps of this technique that we've developed, starting with the remote sensing side of things. Right after I tell you about the island French Polynesia, which is where we did this study. This is an island out in the South Pacific. It's the island right next to Tahiti. And this is where we have an LTER, a long-term ecological research site, that I was really fortunate to get to work at for several years. But I didn't necessarily get the good years. I got there right about when this happened. A marine heat wave that struck in 2019 and killed about half of the living corals and left behind this standing stock, almost a continuous canopy of dead coral skeletons. So our first goal here, once this happened, is we needed to come up with a way to really efficiently and accurately quantify how much coral was lost during this event. So I'll walk you through this process. And like I said, everything you do underwater is a lot harder. We don't have the luxury of GPS like you folks do on land, so we have to make our own reference system. And this was almost enough to get me out of the business altogether, because this is probably the hardest part of this whole process, is where you actually have to go drill into the reef these fixed reference points that you can never find again when you go back to them. You try to screw in these coded targets. These help with aligning different images to create a photo mosaic. And basically, they create your GPS system, right? It's a fixed set of reference points. You know exactly where they are in a photo. And we have to measure painstakingly the distances in between these different points with a measuring tape over 5 meters to the nearest millimeter while you're getting sloshed around underwater. So again, this is not the funnest part of the process. But once you do that, you have your reference system set and you can go out and acquire your images, which is a little more fun. You get to take a camera and you get to be the drone. You swim back and forth, taking the images of the reef back and forth, overlapping images, hundreds and hundreds and hundreds of photographs. So then with those photographs, you can create a photo mosaic, this is actually a combination of about 300 individual photographs over an area of about 4 square meters. This is one of our little baby plots. And while it might look like a still image, it's actually a 3-dimensional model that captures all the 3-dimensional structure of those corals, which is really important. It really helps us understand how changes happen on the benthos a lot more accurately. So once you build a 3D model, you can create what's called an orthophoto mosaic. So it's just one of those big super photos, a combination of about 300 to sometimes thousands of photographs. It looks like a single image, but it covers a really large area and it has really high resolution associated with it. But again, that can be a really difficult thing to extract data from if you had to do it manually. So in that image up in the top right there's probably about 1,000 coral colonies, and they all have completely jagged, irregular borders. So you can imagine if you had to somehow measure them by hand, either in person or on a computer, it would be a nightmare, and it is a nightmare. But we've come up with these ways to annotate these images using AI to make this process much more efficient. So here's one of our reef plots before the coral bleaching event happened. And these are about 25 square meters. These are like the medium-sized plots. And again, you can imagine it might be a little bit difficult to go through and measure every one of those coral colonies by hand. So I'm going to tell you about some of the traditional methods people use to analyze coral cover in photographs. People used to do what was called point contact, where you basically lay down a grid of points that you can go through and easily classify whatever the cover type is underneath, whether it's coral or sand or algae or rock. And this can be a relatively rapid way of measuring different cover of different organisms of the reef, but it's really coarse, right? You get this very rough estimation of what's actually happening down there. A more accurate method is called manual image segmentation, where you actually take your mouse and, again, painstakingly trace the border of every single coral colony, and it will make your eyes bleed, I promise. And so this is more accurate, but it's really, really slow. Now with our method, and I say our method because I'm emphasizing the software engineers, computer scientists who designed this program. I'm just the guinea pig, but you can train a robot or an algorithm to trace the coral colonies for you in what we call semi-automatic segmentation, which I'll show you in a second. So basically, the AI helps you build a training data set really rapidly so that you can then fully automate the process. So annotating this entire image, every single one of those coral colonies would have taken a human probably 10, 15, 20 hours. But this fully automatic classifier can do the same thing in about 10 minutes. And usually a better job, actually. And because we use that photogrammetry, that remote sensing method to collect the imagery, we actually have digital elevation models. So this is sort of like a topo map of the coral reef that's underlaid underneath the photo mosaic, so that whenever we make measurements on that photograph, they're actually three-dimensional measurements. Okay, so I'll show you a little bit under the hood how this works. So this is the interface of a program called TagLab. It was developed by some Italian software engineers, and it's totally free. You can download it off GitHub. People have been trying to like put all kinds of different stuff into this program to see if the tools work the same way, from like trees to like cracks in walls to paintings. It's a really powerful program. And I'm happy to show anybody a little bit more if they're interested. So say you wanted to measure that coral right there. You can zoom in, and all you have to do is click 4 points around that coral colony, and it will automatically measure the coral for you, identify it, and give you a bunch of different metrics like the perimeter, the planar area, and the three-dimensional surface area. And so that's semi-automatic segmentation, meaning that you just give it a couple reference points and it'll fill in the rest for you. Once you build a training data set using that method, once again, the whole process becomes automated. So all you have to do is click one button and it will annotate everything in it that you've trained it to annotate. So now if we start taking different time points of the same area, we can start to see how the reef is changing. So we were able to collect images of these reef plots before that heat wave struck in 2019 and afterwards. And so those more pinkish blobs that you see more of on the left side here, those are the live corals, and those darker brownish, blackish, whatever color they are, blobs on the right side, those are the dead coral colonies that were knocked out in the heat wave. You can zoom in and you can see that same coral we measured in 2019, unfortunately did not survive. But lots of other ones did. So it's kind of interesting to start looking at different metrics of which corals lived, which ones died, how did the proportions change, and the size structures and things like that, all of which you can get out with this program. So once you kind of hit that automatic annotation button and you get all your corals measured, we want to go through and just make sure it did it well. And when we've looked back at these things, we've seen that for most coral species that are at least pretty common, the accuracy is about 90 to 95% when you compare the automatic classification to what a human classified, so it's pretty good. And then you can start to quantify the impact. So I'll just show you a little bit of data here of what the reef looked like before and after this heat wave struck. So one of the first metrics we looked at was just how did the proportion of live and dead colonies change? The number of live to the number of dead from before the heat wave to after. And you can see there's a pretty modest increase in the number of dead colonies after the heat wave struck, and also a pretty modest decrease in the number of live colonies. But if you actually start looking at this, not in terms of the numbers of colonies, but the actual surface area of live and dead coral and how it changed, you see much more dramatic differences. So we lost about half of the live coral colonies, or half of the surface area of live corals. But we saw over an 8-fold increase in the amount of dead coral on the reef. So this indicates that it was actually the largest coral colonies that were most affected by the bleaching. So the biggest ones died, and therefore they contributed most to adding more dead coral to the reef. And then one last little note about why 3D is so much better. We actually compared how the magnitude of coral loss you would have gotten using that 3D metric versus the 2D metric. So a 2D metric meaning like you have a flat image, you trace a border around something, and you get a two-dimensional flat area, right? That 3D metric we have, imagine that coral surface is coming off the page at you. You're accounting for all of that three-dimensional complexity. So using this 3D metric, captured a much greater magnitude of coral loss than that 2D metric. And this is cool from like a technological standpoint, but it's actually really important ecologically because this is going to allow us to much better estimate the impacts of disturbances on ecosystem structure and function if we really know how much stuff we're losing, how many corals or trees or anything else. So just to summarize a couple of the benefits of this approach. Combining remote sensing and AI-powered image analysis allows us to really accurately and efficiently process high volumes of data over large spatial scales on coral reef systems, which was a really big advance for us. It was just really difficult to do that before. Using that 3D metric that the remote sensing technique allows really improves our estimates of reef change compared to that 2D metric. And when we have better change detection, we have a better understanding of the ecological consequences of disturbance. Okay, so now that I've introduced you to the technique that we used, I want to tell you about how we applied this to answer that question of how that skeleton, dead skeleton material legacy affects coral reef resilience. And so we applied this to an experimental setting. First, I'll give you a little ecological context. Since I'm, again, speaking to mostly a room of land dwellers. Coral and fleshy macroalgae, also known as seaweed, compete with each other for attachment space on the reef and for light. They both depend on light for photosynthesis. Herbivores, like this parrotfish, help mediate this competition. By removing algae from the reef, they actually favor the establishment and growth of coral. So our question was, how does the dead skeleton material legacy, or just how do dead coral skeletons mess with this relationship that's really important for reef resilience? Okay, so we set out to do a really sophisticated experiment where we used really sophisticated tools to remove dead skeletons off the reef. And I gotta say, anytime I show a picture of a hammer and a bunch of coral skeletons piled up, I gotta say those were all dead corals. We didn't touch any live corals. We left all the live ones in place. So after that heat wave struck and all that coral was killed and left in place, we went to some areas of the reef and physically removed it using a hammer and a chisel, and then moved it elsewhere on the reef. And we just wanted to follow how the coral and macroalgae communities developed, either when dead skeletons were in place or when they were removed, sort of to simulate a tropical storm that would remove the skeletons compared to the heat wave that left them in place. So once again, here's one of these experimental plots. This is an unmanipulated one. So I'll come point on that side too. You can see these brown skeletons are the dead corals, sort of interspersed with the live ones. Dead, live. So it's sort of like this mosaic of live and dead coral colonies all smashed in together. And this is what the reef looked like just after the heat wave occurred, maybe 6 months after. And so I'm going to show you sort of a photo time series here of how the reef changes, or how the reef changed over about 4 years. I'm going to focus your attention in to a couple of spots here. So if we fast forward 1 year, the first thing you notice up in this kind of upper ellipse here is a lot of brown algae starting to develop. And it's specifically growing on top of those dead coral skeletons. And you see a little bit of it down here too, also some purple algae. So it sort of latches onto the dead skeleton first, but then if we keep fast forwarding through this time series, it actually advances further from the dead skeletons onto the adjacent live coral colonies. And if you keep moving forward, you see this algae advance more and more, and the skeletons start to break down until eventually, by the end of the experiment, basically you just had this little coral wasteland full of dead coral, lots of algae, not many living colonies left. I know everybody has gets trauma about coral reef talks and papers and things like that and stories that are all just doom and gloom and really sad. I'm trying to be a force for change here because it actually is much more encouraging. What we found is that when you remove dead skeletons, so here's one of our manipulated plots where all this space in between those living coral colonies is where we physically remove the dead skeletons. And I'll again draw your attention to a couple of spots here. After a year, we saw some of that macroalgae come in, but it's a little more exposed and therefore is more accessible to those herbivores like the parrotfish to come in and remove it off the reef. So that by a year after that, it was mostly removed from the reef, and there were lots of healthy corals still on the reef that continued to grow and flourish all the way through to the end of the experiment. So just to show you that I'm not cherry picking those two plots and photos, I'll show you some data. So when looking at the live coral, so here I'm showing you the plots where we removed the dead skeletons on the left and the ones where we left the skeletons in place on the right. And this is how much live coral there was on average in those plots after 4 years. So where we had removed the dead corals, there was 60% on average more live coral than in the plots where we had left those dead skeletons intact. Now looking at macroalgae, it's just the opposite. Where we had left those dead skeletons intact, there was more than twice as much macroalgae specifically growing on those dead skeletons. And so we wanted to look at that relationship a little bit deeper. Like, what is the role that the dead skeletons play in facilitating macroalgae? So we just sort of looked at this relationship, macroalgae cover as a function of the amount of dead coral in a given plot and in a given year. And there's a strongly positive relationship. So when there's more dead coral, there's more macroalgae. And this is something that hasn't really been looked into that much before, showing that the dead coral is actually serving as this alternate type of substrate that's facilitating macroalgae. Then, if you look at the relationship between how much coral was lost and how much macroalgae was present in a plot in a given year, we saw that when there was more macroalgae, that drove steeper losses of coral, and the dead coral is what was facilitating that macroalgae in the first place. So again, the benefit is that you remove the dead skeleton, you basically remove that substrate for the macroalgae, and you give corals basically a better chance or a better foot to actually recover after a disturbance. So moving forward, I want to start digging into this data set that we have a little bit more deeply and doing some more spatial investigations. So here's one of our reef plots. And here is the set of automatic annotations, live and dead corals. You pull out those annotations just as a shapefile. And then you can start looking at things like neighbor dynamics. So which corals have which neighbors? So you can draw a border around each one of those live corals, and then you can start to measure and estimate how much of that coral is sharing a border with a dead coral, and measure the degree of overlap, and then look at growth and survival of corals based on how much overlap they have with adjacent dead corals. So this is sort of the next step in terms of what we're doing. We always want to look at how macroalgae sort of advanced through a plot over time. But just to come back and answer this question, dead skeletons are bad for reef resilience, at least in what we're finding. But more encouragingly, removing those dead skeletons can sort of mitigate some of those impacts and cut your losses as far as continued coral decline after a disturbance occurs which might actually prove as like a promising strategy to manage for coral reef resilience. All right, so now I want to tell you about how I'm taking some of these, trying to answer some of these same questions in other ecosystems while I'm here at EASL. So many people say that corals are just the trees of the seas. I'm just kidding, nobody says that. I'm the only one, but I'm trying to get it going. So just as corals serve as these foundation species in coral reefs, foundation species are abounding in all sorts of different ecosystems. And therefore, when they die, they leave behind material legacies, like on oyster reefs, they leave behind dead oyster shells, or in forests, they leave behind dead standing or fallen trees, we call snags or logs. And in salt marshes, there is dead salt grass, marsh grass hay, all of which have different effects on different processes or conditions that are that influence ecosystem recovery. Therefore, these different legacies might have different varied effects on ecosystem resilience. So what I want to do is explore this over a whole array of different ecosystems, ranging from the tropics up to the Arctic Circle, from marine systems to terrestrial systems, leveraging the LTER time series, or the LTER network. So this is a whole system of different research programs that collect time series in very similar ways, but in very different ecosystems, and have long-standing records. So I'm looking to compare different metrics of ecosystem resilience as they relate to, or as they're influenced by material legacies. Just so for a couple examples here, I started doing some preliminary data exploration on some of these different data sets. This is Kansas Prairie LTER, which is a tall prairie grassland out in Kansas. And I looked at how productivity of tall grass prairies is affected by the amount of dead biomass that's in a given plot, or the amount of dead biomass that was there the year before. And it's just, I would take this trend line with a big grain of salt, so I have to do a bit more to verify that relationship. But so in this system, this is an example, another example maybe where the dead biomass is reducing the amount of productivity, right? A negative relationship between dead grass and productivity the next year. We look at another ecosystem, the Virginia Coastal Reserve, which has both oyster reefs and seagrass beds, but specifically for oysters here, this is the density of new recruiting oysters as a function of the density of dead oysters in the previous year. And this is a well-known relationship where the material legacy actually has a positive effect on metrics of ecosystem resilience, like this colonization of new individuals. So this is sort of where I'm at with this project, is just exploring these different data sets. I've been talking to researchers from, I think, 12 different LTER sites and all these different ecosystems, trying to get my head around all the different dynamics from all the different systems, and trying not to mess anything up or piss anybody off. So this is one thing where I am happy to have anybody help and collaborate in any way they want. So let me know if that's something you might be interested in. And then my other project that I'm starting to spin up here at EASL is a little different, but similar to my work, my remote sensing work on coral reefs. And it's looking at spatial patterns of tree mortality at neon sites. So there's this really cool program, data set that was developed by Ben Weinstein's group using neon sites, where they basically automatically detected and set bounding boxes around different tree species at all neon sites. And so they have a data set of 100 million individual tree crowns. But what I would be really interested to do is actually take these from bounding boxes and turn them into more of those, I guess they call them irregular tree crown delineations. So combining this data set with this algorithm that's been produced by other people to try to look at dynamics of dead trees and live trees at neon sites and explore things like is mortality clustered after a certain type of disturbance, or is it more dispersed? Does it advance after the initial disturbance occurs, and things like that? So akin to some of the questions I was asking in the coral reefs. So at this point, I've been talking for a while. You might be wondering, why does any of this matter? So material legacies of foundation species are becoming these really prominent fixtures of contemporary ecosystems. And so it's going to be really important from a management or conservation standpoint to understand how these structures influence resilience, especially if these structures are the products of novel changing disturbance regimes. So in some systems, they're already using and manipulating and leveraging material legacies for management purposes. In forests, prescribed burns are used to get rid of some of that small woody debris to mitigate the risk of severe wildfire. And then on oyster reefs, material legacies, or those dead oyster shells, are actually deployed to facilitate recovery of the oyster reef. So I think material legacies should really be incorporated much more widely into management plans of ecosystems in general. And that's sort of what I'm hoping to get at with my work here. So I think with that, some funding, some people to thank, and then I'll take any questions. Thanks for listening.
Why were the more voluminous corals more sensitive to the heat? Sort of seems counterintuitive a little bit.
I think that's what we're trying to figure out. A couple ideas I have for that. They have more surface area than a smaller one, so they have more surface that's exposed to warmer water. They tend to be higher up, and so they are basically more exposed to sunlight, whereas those tiny little ones can kind of like seek a little bit of shade and shelter underneath them. It also is turning out that things that we thought were the same species are different species. So you might see a small one that survived and a big one that died that you thought were the same species. Turns out they're just different species with different growth forms, but you can't tell unless you have genetics to do that. So I don't think that answer is like, or I don't think that question is completely answered at this point, but those are some of the thoughts that people are having at this point.
Well, I love the experiment of removing the stuff, but you obviously were pretty selective, and that was pretty manually intensive to do that. If we're thinking about like the entire island of Berea, can you drag and get rid of this stuff quickly and then recolonize with the live stuff, or do you have to go and be really careful about pulling out just the snags in between everything?
Yeah, this is the thing that is like really important to think about, like how to actually scale up this process or this technique. Yeah, you probably don't want to just go troll the whole bottom, right? Because there's still going to be a lot of stuff that's living down there, even though that's technically what a cyclone would do. I think it would probably be a little better to, be selective. But I think probably the case is that you don't have to go remove everything. You just got to remove a little bit so that the algae doesn't have this continuous surface to expand over, right? It's almost like creating like a fire break, right? You just kind of you draw a line in the sand or on the reef, just create space between dead coral heads so that algae can grow on a coral head, but it can't advance any further off of that. So I think it's going to be a little bit more about not how do we remove all the dead coral, it's like how much dead coral do we need to remove and in what spatial configurations and how much of the density and standing stock do we need to reduce before we get these like beneficial effects.
And then the parrotfish, do their population numbers not increase when you get all that algae, and so then herbivory pressure eventually goes up? Do we not see cycles in that?
So after the cyclone, there was a cyclone in Maria in 2010 where the reef was kind of just totally leveled out. Algae started to creep up and like increase in abundance. Then you saw this huge response by the herbivorous fish community. And then they came in and knocked all that algae out. And then you saw this recovery of coral afterwards. After the bleaching event, we're seeing all that algae coming up, but we're not seeing the response by the herbivorous fish community, meaning that they don't have access to the algae. The algae's there, they just can't reach it because it's growing in between the branches of those dead skeletons. So they can't access it, they can't remove it. Therefore, their populations can't increase and they can't control the macroalgae that's growing there.
Are there serogens in the system, and like what's the potential for like remotely sensing like sessile herbivores like that? Because I know that's important for like regime shifts and that sort of thing.
Yeah, I wish we had sea urchins. Maybe that's the answer, is just bring sea urchins. urchins are pretty minimal in this system. You'll see them kind of like they're like little Roombas just kind of going around like the dead coral heads and up and in between the branches. So they would be really great and actually really good bioeroders of the dead skeletons. So that'd almost be like a nature-based solution to an urchin. Well, I mean, anytime you introduce one thing, yeah, lots of other consequences. But there are native urchins there, so maybe putting those in places where there's lots of dead skeletons could actually be really beneficial. Remote sensing them, yeah, I mean, sometimes you look at these the ortho images and you'll see a fish just sitting right on top of a coral. So if we can get those, I mean, I'm sure we can get urchins that are moving a lot slower. As long as anything's just sitting still enough, it'll get picked up. So yeah, maybe not reliably like for counting urchins, but you know, yeah, they'll get picked up. Yeah.
So in all your plots, how heterogeneous is the structure that you see across those plots? It seems like where you start matters a lot with potentially these feedbacks you're seeing and leaving the material guide.
You mean like
Like, you know, really dispersed coral
Yeah,
versus
exactly.
more dense coral, and how might that affect your interventions?
Right.
Yeah.
Yeah, so okay, so this is a Pacific reef, which tend to have a lot higher coral cover. And in this case, like the way the reef looked right before this heat wave hit, it was just like you couldn't see the reef, it was just coral, all completely packed together. It looked like a forest canopy. But then when all that coral died, it created a really dense and continuous canopy of dead coral. So that kind of situation, yeah, is concerning. And I think that was probably mostly the case for these plots that we had, you know, because they were on like the side of the island that had the most coral before the heat wave hit. But if you go out to the Caribbean, you don't really get that density of coral to begin with. So the same consequences might not even really matter here, right? Or in the Caribbean, I mean, where you have corals that are so spaced out and there's like hardly any coral there to begin with. Yeah, you're totally right. So when you have like less dense packing of corals, these issues probably won't be as important. So yeah, it depends a lot on how packed in it is and how homogeneous that canopy is.
I loved all the coral reef stuff, really interesting.
But
I'm gonna be the person that is asking about the grassland plot that you're working on now. So I'm just curious, when I think about grasslands, there's a lot of these other ecosystems that you're working on. Grasslands senesce their grass every year, so that standing dead is gonna be there every year predictably, versus, you know, coral reefs or trees, it takes a disaster to cause that standing dead to be there, right? What do you think the impact of that is going to be? You know, a cyclical process of it, or more once in a, you know, decade type cycle or something like that?
in grassland.
Or just in general, you know, how grasslands might respond to that standing dead versus other ecosystems.
I guess with the grassland, I think about got it. Konza Prairie, they have this whole long running experiment where they do different frequencies of fire burning.
Yeah.
And so fire frequencies are changing. That's like one of the ways in which disturbance regimes are changing there, right?
Sure. Yeah.
So when you have really frequent fires, it reduces that standing dead biomass more frequently, which I'm told is good for the system, right? It makes things more productive. But if you all of a sudden have like fires that are happening much less frequently and are more intense, then like you might, for one thing, get a huge buildup of standing dead, which reduces productivity. And then you get a really intense fire that actually like burns down to the roots, and therefore you get slower recovery too. So it's like, once again, thinking about how the changing disturbance regime is also changing the material legacies and their relationships to the foundation species.
Okay, interesting. I like that.
Does that make sense?
Yeah, no, totally.
Did I pass?
I wasn't a quiz. Which fire regimes are you looking at? They're all of them, or?
I am looking at all
Yeah,
of them.
okay, cool.
And I'm pretty sure what I showed you was all of them. I'm going to start parsing it out a little bit more. And this is kind of like the step is like, I tinker around with the data for a little bit.
Of course, yeah.
Until I reach a point where I'm like, I have no idea if this is right or not. And then I go talk to the experts and I say, is this right or not? And they'll tell me it's not. And I'll like, reiterate from there. So that's the idea with that.
I love learning about corals. That's really cool.
I love learning about grasses. Yeah.
Thank you for your presentation, and I'm wondering if it is possible to do the 3D mapping using neon and LPR dataset as you did in the first.
It is possible to do what?
So 3D mapping
Yeah.
of the terrestrial and marine areas with different ecosystems.
Yeah, definitely. So I know that with the AOP, the aerial observation platform through Neon, like they're collecting photographs in the same way, basically the same way that we were collecting photographs on the reef, just like from much higher up and covering a much bigger area. I was so jealous when I first found out how much area they cover in like with a single drone flight. I'm like sitting here like this, and then they're saying like, oh yeah, I've got like 40 hectares in one flight. Right. So anytime you're doing mapping that way by taking continuous overlapping photographs, you're able to can reconstruct the three-dimensional structure of that. But then they also can do it with LiDAR, which is even more impressive, I think, because you get different canopy levels and the structure below. And so, yeah, I mean, that's the idea, is that they have the same kind of like photomosaic imagery that can be overlaid on top of this like very structural map. So I'm trying to figure out exactly how much has been done with that, and if I can find a little way to sneak my way into the terrestrial community here and try to show or use some of the same techniques for just like actually tracing the whole tree crown the way we did with corals and measuring the three-dimensional surface areas of them. Thanks, everybody.